Next up is, and I have noted the time, so you'll still get your full whack, don't worry. Lisa Stubbs is going to talk about a conserved regulatory core with species-specific innovations at the root of shared behaviors. So genetics and behavior, interesting subject.
All right, so first of all, it's a pleasure to be here. This has really been fun so far, and I'm looking forward to the rest of the conference. I'm going to tell you about a project that I'm sort of a newcomer to in a lot of ways, and I'll explain that in a moment, that blends together interests from a broad range of individuals that I'm collaborating with and has got us all very excited. So let me start with, oops, I'm giving the wrong signal. There we go. So first of all, let me just talk about my own background, my own genotype. My background is in transcription factor evolution and regulatory network evolution. So I'm really interested in how genes are regulated and how those regulatory mechanisms change through evolution, and especially in the context of development. So I've been really interested in brain and reproductive development in particular because they change a lot in evolution. And that's what my own research program has been based on for many, many years. But since about 2008, when I moved to University of Illinois, my interests have been somewhat tempered by my environment. In particular, I moved to this wonderful place called the Institute for Genomic Biology at UI. And the idea behind IGB is to co-house people next to each other in the same floor that have really different affiliations, departmental affiliations, and really different kinds of expertise, and get them to work together to kind of think outside the box in terms of coming up with projects that they would never do on their own, but they might be able to do together. So in my particular team that I was recruited into, it's a lot of computational biologists, there are physicists, bioengineers, there are some other developmental biologists, including myself, but also I got brought into a cluster of people who are neuroscientists and with some particular interests that wanted to try to adapt some of the information that we've developed over the years about gene regulation during development and its evolution over history, and try to bring that into the realm of behavior. And that's the project I'm going to talk to you about today. So the people I'm working with work with a wide range of model species from insects to mammals, and that's quite a broad range. And that's part of the thing that makes this also challenging and interesting to us. So rather than just thinking about your inner reptile today, let's think about your inner honeybee, all right? Your inner insect. All right, so the basic concept is this, that despite all the real big differences between, say, a fly and a mouse, there are underlying mechanisms that operate very early in development that are extremely well conserved. So the outcomes are really different, right? This and this have almost nothing in common unless you really think down to the deep level, the cellular level. But if you think about the organismic level, they're really, really different organisms. And yet, there are very, very conserved networks of genes. And so what I've drawn here, just because it's a nice illustration, is the notch network in Drosophila, which is really well characterized. And it turns out that there's a network very much like that in mice. And it operates very similarly in development. It's involved in early differentiation decisions, been recruited into many different kinds of tissues to have particular outcomes, but operates pretty much the same way. The same sorts of genes are talking to the same sorts of genes in this network. And that's something that's been conserved over all these millions and millions of years. Now, there's some interesting innovations that get added onto this over evolution, of course. And one of them is that mice have about, well, it's not quite, it's about, there's been a quadruplication of the genome between insects and mammals. That means that for every one of Drosophila's genes, we have about 4 of them. Now, that's not necessarily true for every gene. For some cases, we only have a 1-to-1 relationship. Some cases, we have 1 to thousands, hundreds. But for the most part, it's sort of an average of 1 to 4. There's been a quadruplication of the genome. And these new copies of Notch, for example, that you see here in the mouse genome, Notch 2, and Notch 1 is in here somewhere. No, I guess this is just Notch 2. But anyway, there's Notch 1, 2, 3, 4. and it has partners that have also been duplicated. That gives us an additional range to this ancient mechanism. So it's the same mechanism, the same interactions, but deployed in different ways in different tissues with some subtle variations on the theme. So that is a very commonly accepted paradigm in development by now. When I first started graduate school, it was still something that was kind of new. Now it's pretty well accepted that when you start thinking about development, you can imagine, in fact, it's very well established that there are genes that do the same job in flies versus mice pretty much at the same sorts of times, the relative times in development. This is the Hox clusters and the Antennapedia and homeobox clusters in Drosophila that have been quadruplicated again in mammalian species, but have the same sort of relative expression across the organism during early development and carry out really similar mechanistic jobs. And so the idea is that a lot of these old bits of machinery have stuck around and then tweaked around the edges quite a bit and are deployed over and over and over again in different ways under novel circumstances and in different combinations so that you end up with an organism with more complexity, more cell types, for example. And that's something that's been pretty well established. Let's say the homeobox clusters are involved in the segments of insects and the somites of vertebrates, so the things that give rise to your backbone, your spinal column. It gives rise to wings, limbs, and fins, all by different tweakings of those basic mechanisms, but it's the same basic molecular machine. And there have been some really interesting innovations to that. For example, in addition to these ancient developmental jobs, the Hox clusters, Hox A cluster in particular, has been repurposed in adult mammals to make it possible for females to get pregnant. So the uterus is primed by Hox A genes being reactivated in the reproductive tract of mammalian females. Now, that tissue didn't exist before. And here it is, this mechanism being repurposed for this completely new job. And that is because there have been redirections of this machine so that it's reactivated in this particular place, in this particular time in mammals. And so there are species-specific tweaks that are added to activate these mechanisms at different times and places. Very well accepted paradigm in development. There's lots of different examples of this. But so let's try to think a little bit further away. It's already accepted for development. What about other kinds of traits? And in particular, my colleagues were interested in the idea that there may be something like this, some sort of conserved machine that underlies conserved behaviors. This is kind of, to some people, kind of a crazy idea because behaviors are really species-specific, right? But there are some behaviors that are very, very specific. I mean, very, very conserved. So there are both deeply conserved and species-specific aspects. And for example, I'll give you the example that we have actually studied so far, which is the response to territorial threat. Now, when you think about it, just about every animal has a territory. And when a conspecific, that means a creature of the same species, comes into that territory and threatens them, or decides to take its food or its mate or something, there's always a common response. It doesn't really matter what kind of animal you're looking at. The response will be very, very similar. And so in this particular case, what we decided to do is to have a look at 3 model species. We chose these 3 species, quite honestly, because they were there. They were there available to us, but also the 2 of the model species, these, the worker honeybees and the male stickleback fish, are actually very well-established behavioral models. There have been lots of publications and lots of research done on the basic behaviors of these species. Mice are not the greatest behavioral model. People would rather use rats, probably. But they're really great genetic models. And we have wonderful genome sequences, lots of resources. And so I'm not a behaviorist, but I'm a mouse geneticist, and I was able to contribute to this project through my mouse genetics expertise. So what we decided to do with a small amount of money that we managed to squirrel away in our theme project, which at the Institute for Genomic Biology, is just go for it and give it a try and see if we could detect any similarities in gene expression and gene regulation patterns across these three species in response to a very analogous threat. So our reasoning was, at very least, we end up with 3 independent papers on the 3 species that we can publish. But at best, what we'll do is we will have some red meat to throw out to our computational colleagues to challenge them to think outside of the box in terms of looking for traits across such huge evolutionary divides, despite all the challenges. And so that's basically what we decided to do. And so one of the questions was, these responses are remarkably similar. Is that just convergent evolution? Or is it a species-specific layer on very ancient basic brain response? And so that's what our question was asking. So when we think about these 3 species, we've got lots of issues. They have really different advantages in studying social behavior, as I mentioned. And one of the things we were really motivated to do in the study, beyond our interest in the 3 species and beyond just thinking about this possibility that there might be some conserved elements here, is how can we really truly leverage all of the information that comes from these disparate model systems that are being studied for biology of all different kinds, lots of different kinds of biology. You have studies being done in flies and honeybees and stickleback fish and different kinds of sooner or later, there aren't very many model reptiles yet, but sooner or later reptiles, there are going to be birds, there's songbird studies. How do you actually really leverage the information you get out of these disparate species to make some sense that you can take home and use when you're interested in human behavior, for example? So that was another one of our motivations. But there are several strategic issues. So which brain regions do we choose? These are the 3 brains here. That's a honeybee. We don't have a mushroom body. And we don't have an antennal lobe. So, what do you choose to compare to this mouse brain here? This is the fish brain. It's pretty weird looking. There are some ways that you can lay parallels between the mouse brain and the fish brain, but it's not very straightforward. And in fact, not very much is known about the fish brain. But for the mouse brain, what we have is a really excellent map of where the various regions are, and we're able to go in and we're able to carve out pieces of the brain using pretty standard and pretty well-developed techniques to dissect the brain up and get particular brain nuclei, brain regions, so we can actually think about testing particular regions of the brain. So the question is, how do we decide which region to connect, to compare to which? The other problem is this one I told you about the genes, okay? There isn't a one-to-one relationship. You can't just say, okay, I'll go take a look at the honeybee gene that is is the ortholog, the equivalent, functional equivalent of my mouse gene. There's usually going to be a one-to-many relationship. There have been lots of different gene duplications and deletions and so forth over evolution. And so figuring out exactly which genes to compare to which is not a trivial problem. And then the question becomes, how do you really calculate, how do you really compare across 3 species? That's not computational methodology or mathematical methodology that's extremely well characterized yet. And so there were lots of different challenges to this. And so what we decided to do in our pilot study, which is all we have data analyzed for yet, was to take whole brains from honeybees. And later studies that we've done now, we've used something called the mushroom body, which I showed you before, which is supposed to be their sort of higher processing region. In our pilot study, we also took diencephalon from sticklebacks. That's kind of a general big region that includes a lot of the regions related to hypothalamus, thalamus, lots of emotional sorts of processing centers equivalent in mammals. And in mammals, in the mouse, what we decided to do is just dissect the living daylights out of the thing, make RNA, and test for the activation of what's called the immediate early genes. So basically, whenever neurons fire in a region of your brain, they set off a cascade of particular genes that are a signature of activation. And so if you can catch them when they go up quickly enough, and they do stay on for a little while, you can catch those what are called immediate early genes, and you can test the entire brain and figure out which is the region that has been activated the most. So you can end up with a dissected cluster of neurons that have been activated more than other parts of the brain. So that's what we did for mouse. Then when we came to considering which genes, we decided to spread our net a little bit and to build a database of genes that are not just these one-to-one, the best possible match to the mouse gene in honeybee, but to open the door and allow for these gene duplication and deletion events. And that's something that several people in our group have a lot of experience in, including me, and so we were able to build that database. Across the Cal3 species significance calculations are a really complicated issue. We certainly are not done with it yet. We're just trying to get everybody stimulated to think about this. But for our first study, we came up with a rank-based statistical methods that combine p-values across the genes and then rank them after comparison across the 3 species. So that's a long story, but let me just tell you what has come out of what we have so far. So, yes? Yeah, the fear response. So, yeah, so the fear response was the resident intruder test for mouse, and so I'll just give you that as an example. It was something very, very similar for the 3 species. For the resident intruder test, what we do is we have a mouse. He's established his home nest. He's had a female in the nest with him. He's made a nice little comfy place. We remove the female briefly before we do the test, and we drop in a male mouse, and this is the first time either male has ever seen any situation like that. They've been in their own cages. This is their first experience to some sort of territorial intrusion. The immediate response is, of course, aggression on the part of the resident male. In the honeybee, there was a much more complicated story. They take a piece of honeycomb, they move it over into an enclosure, they have bees that become to recognize that honeycomb as their home territory, and then they bring in intruder bees. These are all worker bees. In the case of the sticklebacks, the males build the nest and the males defend the nest, and so the males have defended their nest, they've built a nest in an enclosure, and a male stickleback is dropped into the cage. So it's very, very similar sorts of 3 species tests, and they're all pretty well documented, established behavioral tests that we modified somewhat in the case of the honeybee and the fish in order to fit what is really established for mice. So then what we did, of course, is we did these dissections, and we took these brain regions and sent them off for RNA sequencing. And so what we're comparing when we do look at the genes and we look across the species is gene expression, gene activation, or silencing in response to the threat. So we're looking at control animals, and then we're looking at treated animals, and we're looking for things that change in a short time period. So I'm not going to go into this in great detail. This little plot is something that I'll explain in a minute. But the first thing you do when you get these gene responses is you end up with a list of genes. So we have genes that are maybe, in the case of mouse, it was about 1,000 genes that had significantly changed. We used the standard methods. In the case of bees, it was about 300. In the case of fish, it was about 1,000. But when we do this cross-species ranking, we ended up with about 100 genes that we could consider to be very consistently activated or repressed across the species that were either one-to-one orthologs, so the same gene across species, or one of these duplicated paralogs. And so we collected all of those genes. And the first thing you do with the gene list when you're doing gene expression studies for genomics is you try to understand what functions are represented by those genes. So there are databases of carefully annotated functions for genes that we can send to and look for statistical overrepresentation of particular functions in the gene list. So this is a long procedure. I could give an entire lecture on how you do those processes, but I won't do that. Just suffice it to say that there are databases that allow us to quantify or to look for overrepresentation of genes that are associated with one function compared to another. And so when we just looked at the one-to-one orthologs, the genes that are strictly conserved between species, one copy in bees, one copy in mice, we end up with some categories that are not that surprising in some ways. We end up with this particular category, which is response to oxygen, which makes a lot of sense in this particular case. But it's also very likely to come as a category, as a really strong category, because those genes are the ones that are represented very highly in these very highly conserved, not changed over evolution genes. Response to oxygen species is what happens in cells that have been activated, have a lot of metabolic activity, and for example, the mitochondria are turned on. So most of these are mitochondrial genes. We also see things like structural constituent of the ribosome, and this is another mitochondrial signal. Those are things that come up when you only limit things to the one-to-one orthologs. But when we open it up and look at this whole spectrum of potential genes, we get a lot more genes, and we get some interesting looking categories that are a little bit more informative. And so one of them is developmental functions. Actually, we saw that in both places. This is pattern specification process. It has to do with early development. We also see signals here of cognition and behavior, which makes a lot of sense. We didn't see those before on this side because those genes are typically not one-to-one conserved. We start to see, we still see, transmembrane transporter activity and so forth that are closely related to neural activity. And we start seeing things like steroid hormone activity and other functions. Now, these are clusters of functional categories, not individual functional categories, but this is just sort of giving you the general types of categories that we saw when we did this analysis. So when we go and look at the particular categories, and we look at the mouse genes that are falling into this cutoff list that we get when we compare across the three species. So we first see some very general descriptors, protein transport, muscle contraction, what's that doing there, hormone activity, it sounds very generalized, and other kinds of functions. But when we go and look at those mouse genes, we start seeing a little bit more specificity just because there's been a lot more research done on mouse. So there's a lot more literature to tap into in these functional categories. So we see obvious things like neuronal transport to the synapse, splicing, translation. Been a lot more activation in those genes, those cell types. They've been flipped on. Their metabolism has been turned up. We see things like neurotransmitter receptors. But also, when we think about the muscle contraction, which was a puzzle here, we suddenly see vascular smooth muscle function and blood pressure regulation. So we were talking about this before. That's one of the first things that happens when you activate a brain region. We now have groups of genes that are associated with this function, and we can see it on a molecular level as well. It's one of the first things that happens is the blood pressure changes, and the vascular smooth muscle in the lining the blood vessels, the activity is very obvious in the molecular pattern. We start seeing genes that on the geo categories in the functional databases, muscle development, which doesn't make a lot of sense. But when you look at those genes in mouse, they're involved in dendrite pruning and dendrite development. That's when the neurons connect to each other and are sending out processes to connect to each other. And so that's obviously a process that's occurring here. And then we start seeing hormone activity. We start seeing not only the steroid hormones, but neuropeptides. And the neuropeptides in particular in the mouse, all three databases come up as one of the very highest activation signals. But those genes are not conserved. Those are lineage-specific genes. So they don't really have correlates exactly with each other. The insect neuropeptides are different from the mouse neuropeptides, and yet in all three species, neuropeptides is the first signal. That's really what's setting off a lot of the follow-on signals that we're seeing in our data set. We also see a really interesting signal of neurite outgrowth and dendritogenesis, and also something to do with cell division, which was kind of a surprise for all three species, but it comes up very strongly, cell division and neurogenesis. And let me just talk to you briefly about what that means. So there's the neurogenesis signal we see has to do with brain development, lots of things that are involved in brain development. Some of them have to do with just what neurons do when they're differentiating. They send out these processes, as I said, and build new synapses. But also, there, as I said, there's a very strong signal of new neuron creation. So it makes sense in mouse because the region we ended up sequencing was the hypothalamus. And in the hypothalamus, it is one part of the brain, in the mammalian brain, that is known to undergo neurogenesis in response to certain kinds of stressful challenges. And so there are some cells called tanocytes that are involved in regulating metabolism in the hypothalamus and also throughout the brain, and also sending out some of these neuropeptides. But they're also radioglial-like, which means that they can become new neurons. So in the mouse data set, we have a signal of new neurons being created. That wasn't known for the other species and has never been investigated in them. But what our three species data set suggests to us is that there may be tanocyte-like neural glial cells, precursor cells in the insect and fish brains as well, and that they may be contributing to this signal. So what that is telling us is that possibly there is some, this first stimulation of this stressful situation with the animals is setting off a new round of neuron differentiation and potentially new neuron creation that evokes some of the same transcription factors and signaling molecules and other kinds of molecules that are best known for their role in development. So we see this very strong signal of pattern specification and neural development that is probably reactivation of these ancient developmental genes for these processes of creating new synapses and creating new neurons. So that's what our hypothesis is. There's lots of evidence that this is probably true in the case of the hypothalamus in mouse, and now we have some evidence that it may be also true for the other species, and it's something that we're interested in digging into. So we found one of the things that we were all very interested in is what is controlling all of this reaction? Well, as I told you, one of the first things that happens is the neuropeptides stimulate some activity in the cell. So that's obviously what sets off, for example, the blood pressure control, the signal of vascular activity and so forth, probably set off just by the neuropeptides themselves, a very fast reaction. But that process is also somehow triggering the activation of a cadre of transcription factor genes. The transcription factors, as many of you may know, are those proteins that regulate other genes. So there's immediate connection between this early response from the neuropeptides and somehow this activation of transcription factor genes. If we look at the ones that are represented in this group, these are the immediate early genes up in this little cluster here. There is some evidence from the mouse databases and so forth that these guys talk to each other, are connected to each other, are known to interact with each other. That's what this drawing shows you. It's a network drawing of established interactions between these transcription factors. EGR1 is one of the classic immediate early genes. It's obviously upregulated, and because we tested for that one in order to be able to sequence. But what this shows us is that this little cadre of genes is connected through at least one of the other conserved transcription factors called SMAD3 through to another little group down here. These transcription factors are known to be involved in neurogenesis, and including adult neurogenesis and new neuron creation in mice. So there is a direct connection between this early activation set and the signal we're seeing in neurogenesis and neurodevelopment that we're really interested in exploring. That's from the existing data from other systems. We're seeing it now in our data set as well, and we're very interested in following this up. So they're known to be interacting in mouse from other experiments, and permits a hypothesis about how these processes may be triggered and then translated into action over time, and so we're really interested in that. So we're interested in a couple of things. One is, where do we find those regulatory elements? I won't go into this in a lot of detail, but this is one of my major interests, in that the regulatory elements that are responsible for turning these genes on can be located far away from the genes. And so that's because our chromosomes are not organized as a linear structure. It is in a three-dimensional structure that's packed into the nucleus in a very specific folding pattern. And similar positions in the nucleus determine how things interact with each other in the nucleus. Because of that, elements that control genes can be really far away from them, and they're very hard to find in mammalian systems. But we have ways of identifying those by using markers of open chromatin. There are some very new methods that allow us to find those places in the genome that are open for business. Those are where we can find the elements that control genes, and watching them open and close over time can give us some idea where the elements are located for particular genes, and that's something that we're in the process of doing now. So in other words, we're looking for not only the genes that go up and down, but the elements, the non-coding elements in the genome that may be quite far away, that are telling those genes to go on and off in response to this neural stimulation. And so we are in the business right now of just analyzing these data sets, and have some good candidates, and are really interested now in trying to discover how those genes go up and down, and how they are regulated in response to this chain of events that starts with the neuropeptide story. So I won't go into this very much. We're in the process of looking at additional brain regions and individual animals. We're trying to integrate across the data sets. This is requiring the invention of some new computational tools and some new ways of thinking about the data that we haven't been really invented yet, that we are going to have to apply for the first time. And that gives us a lot of exciting things to work with. So I just wanted to acknowledge our team. This is a multi-group effort, as I mentioned before. These are the individuals that I'm collaborating with on this project, and we're funded by the Simons Foundation and by NIH and NSF individually and as a team to do this work, and are interested in trying to press further into the mechanistic details of how these things are organized.
Thank you. So questions. Can I, if I take chairman's privilege, can I ask you a question? Some of the genes that are getting switched on seem to be involved in somewhat longer term activity than you might expect from the immediate threat, like neurite outgrowth. Why do you think that you're getting the possibility of growing new neurites when it's a challenge that you think will be over in 2 minutes, will fighting the other guy off? Is this getting ready to some big defense that they think they're going to have to do for the long term?
Yeah, I think this is a learning experience for these animals that they're obviously sort of primed to do. They have never been in a situation like this before. The time period that after we test the animals is very short after the exposure to the threat. On the other hand, it's long enough that at least the genes that are going to be involved in sending out neurite processes could be turned on. That doesn't mean the proteins are there yet. Right. It doesn't mean that they've been localized yet. It doesn't mean that they've been activated yet, but the genes have already been turned on. And that's the signal we're seeing since we're just looking at RNA.
Good.
In fact, we don't see the proteins activated that quickly.
Sorry, I was going to say guy in the green T-shirt.
Right. We don't have enough money to study both yet, and so we're just trying to keep it a little bit tidy so that we can really just look at the defenders at the moment. But we are interested and we are saving those animals. We just haven't analyzed them yet. Yeah, we tested a lot of regions, and the signal we saw at this particular time point was hypothalamus. Later, we may see, so one of the things we're really interested in doing and looking at the later time points is watching both by in situ hybridization and so forth, and by RNA-seq, and by qPCR and so forth, to look for what's happening to the activation pattern. How is it tracking through the brain? And at what time course, and in what regions is it going next? And so that we can kind of, we haven't got that data completely yet, so we're just in the process of analyzing that data. But we're interested in all regions that are activated.
Oh, no, sorry, in the back. Go, you had a question, did you not? Far away.
Yeah, so defining behavior is probably an interesting question here. Some of the behaviors that you're describing seem like really generic stress response or muscular response or something. So I wonder how you think it might generalize to other things that when we think, wow, how could behavior be conserved between different creatures, we might be more surprised? I don't know. I know nothing about this
Certainly,
field.
if we look at other behaviors, we're going to probably see different kinds of signatures, but I think it's mostly going to be different signatures or similar signatures in different regions of the brain. That's what I think. Now, I don't know that for sure. I think what we're seeing is sort of a generic ancient signal machine for what happens when a region is activated and a memory has got to be set. This is clearly one of those behaviors. We chose it because it's so obvious. It's not subjective. Right. Yeah, we just know exactly what the animal's going to do, and we can tape it, and there's no question that the reaction is the same. Later on, it would be really interesting to get into some more complex behaviors. We just wanted to start here so that we could develop all the tools for doing this cross-species analysis, which they're just not available yet.
So the chairman was in his privilege, took my first question, but I'm going to ask the second one. I'm thinking as a fly guy, are there any mice or sticklebass fish or honeybees that are cool with another male dropped into the mix? Sorry, Larry, that's not what I meant.
Well, if males were raised together, like their brothers, or just weaned at the same time and kept in a cage together, and they've never seen a female, they do just fine.
But yeah, so if another male, if they
A
built a
novel
nest,
one, no.
So you never see a mutant of that behavior.
I've never seen. There's probably one. Actually, we probably have one. We have an animal that doesn't respond normally, so socially, it's a mutant that we generated. And they probably wouldn't react the same, but we haven't tested it because
I just think that'd be an
we haven't
interesting.
done it
I
yet.
think this happens in academia. Do you think this kind of behavior happens in academia as well?
Of course.
Last one before tea.
Oh, dear. So this was incredibly exciting to me, actually. And I ended up thinking during your talk that you did what the Institute of Genomic Biology would have done, which is you did next-gen sequencing because you could. And so in a sense, you did a looking under the lamppost experiment that stayed away from the question about kinetics. And if I were you, I would wonder if you're considering something unbelievably stupid and simple, which is to take the pieces of brain that you're going to grind up to get RNA and stick it in buffer and not break the cells and take all the little peptides that come out and do horrors upon horrors, for me, a mass spec analysis of the, that's not funny, the stupid peptides, because there will be some release of something that's kinetically, neurally active right away, just like Tim
I
said.
agree. I think that would be extremely interesting. I totally agree with you. In fact, we really are interested in doing that. It's just a matter of what are we going to, with grinding up a piece of brain? Oh, just, okay. It would be really fun to do something like that, and we've been talking about it. We just don't know exactly how to do it, so we can maybe talk to you about it. That would be really exciting because I think it's not all about gene expression, right? It's not even, we can see the neuropeptide genes go up, so something is triggering them, but there's probably something that was there in the beginning that was a protein, that it got modified or something and quickly acted, and those are the very early events.
Okay, well, just before we break for tea, the artist of the picture that's on the front of your booklet and the bigger picture is in the back there is going to come down and say a few words, I imagine, about the origin of the picture and what it actually means. But like the acting, it probably means different things to different people. And I'm certainly sure that the artist had something in mind when he painted it. So please tell us.
Hey there, everyone. Larry, I'm back. Yeah, this is the third year that I have collaborated with Larry and his team on the artwork for the GoldLab, and every year for me has been an increase in interest, curiosity, engagement, and as I've come to understand a little more of what the symposium is about. And so we were talking about the artwork last night and going into the details of it, and a number of people expressed interest in and curiosity if there was a print. So I have now released prints of this artwork, and the original is right up there. It's a pastel painting just outside. And yeah, a lot of it was based on Daniel Kahneman's Thinking Fast and Slow, as well as a number of other great sources and conversations that I had with Larry and his team. So yeah, it's great to be here, and buy my prints. Yes, thanks, Larry.